Hello friends, today here we are again with a new video of biology and our today's topic is development of embryo. In our previous lecture you have heard about development of endosperm. Endosperm, when we have seen, that time we have seen a triple fusion in between. Here I have written pen, P-E-N. What is meaning of pen? Pen means primary endospermic nucleus. We have seen it is triploid structure. Triploid means N plus N plus N, that is 3N, triploid structure. And zygote is a diploid structure, that is 2N. When we are mentioning zygote, we write here 2N. So, how the zygote is developed? Because when you get the concept of zygote, then and then only you can get the concept of embryo. Zygote is the combination of male gamete with the female gamete, that is N plus N. When we are taking a consideration of male gamete, we write N plus female gamete, that is N. Female gamete is egg cells, male gamete we can say pollen. Together it forms a diploid cell, that is zygote. The development of zygote from male and female gamete is termed as formation of zygote. After all, it is developed in the form of embryo. So the development of embryo together is known as embryogenesis. What we call it as embryogenesis. Embryo is formed after the formation of zygote, and genesis means to form. That's why together the topic is known as embryogenesis. Before we start the topic, we should know about the embryo sac. Embryo sac is a kind of a bag. We know that it is in the female gamete where we have two ends. One is micropylar end and another end is known as a chalazal end. This we have seen in the anatropis ovule, that is micropylar and chalazal. Micropylar end is having the egg cell in between and the synergids on both the sides. Synergids get degenerated after some time, and egg cell when fuses with the male gamete, one of the male gamete, it develops in the form of zygote, and in between we get the pen, that is primary endospermic nucleus, by the triple fusion. And on the chalazal end we are also having an antipodal cells, which are also a subsidiary cells which help in the development of embryo. Later on, this zygote means here the 2N is developed zygote, which is also known as a proembryo. We also call it as a proembryo. It's an immature embryo. After the later stage, the zygote is transferred divided into the two different cells. One is a micropylar cell, that is third one. Another is a embryonal cell. The micropylar cell is the upper cell, which is also known as a basal cell. It is a larger one. And the embryo cell is the smaller cell, which is a terminal cell. Micropylar cell, suspensor cell, after a few transfer divisions, start develops into such a suspensor. That's why we call it as a suspensor cell. Suspensor cells, after some times, after transfer division, develops into a globular-like structure, like a ball structure. Globular, it develops into a formation of embryo. Then, after the transfer division, it reaches a octant stage. What we call the stage as octant stage. Octant stage means 8-cell structure. This structure is common for both dicots and monocots. That means after the transfer division of octant cell, they form a heart-shaped structure. That is a similar structure which looks like a heart. That's why we call this kind of embryo as heart-shaped embryo. Upper side suspensor is formed. I will give you the significance of suspensor. What is the role of suspensor? Suspensor helps to push the embryo towards the endosperm. Means the function of suspensor is pushing the embryo, that is this embryo after formation of zygote, towards the endosperm. Why it pushes the embryo towards the endosperm? For getting a nutrition. Endosperm provides a nutrition for the development of embryo. Later on, the cotyledon, that is together with the hypocotyl and cotyledon starts enlarging. Enlarge means it starts multiplying transversely multiple of times in a right angle division to form this cotyledon shaped structure. We call them as horseshoe structure. But when the horseshoe structure forms, you can see here there are two cotyledons are seen. Two cotyledons means we call it as a dicot embryo. What we call it as? Dicot. There is a difference between dicot and monocot. In dicot we see two cotyledons. In monocot we will see a single cotyledon. In cotyledonous structure you will see that from middle we get a plumule as well as on upper side radicle is formed. Plumule after some time will form a shoot. That is the upper part of the plant. And radicle will develop into a roots. That is the lower part. So we should know that plumule is developed the shoot formation and radicle will help in the formation of roots. That is the basal portion. Together we study the dicot embryo in such a form. So this topic is very important and you have to draw the structures in your notebook in such a way. It's an easiest way to draw the structure, or either you can develop such a structure's ability. The diagram is compulsory when you write the answer. It gives you a additional points when you are representing an answer with an diagrammatic presentation. So thank you for watching my video.